Good morning.
Good morning, madam.
Good morning. Good
Anupama,
morning.
ma'am, please meet our principal, ma'am, Professor Sumakant Garu.
Yeah, hi.
Good morning. Good morning, Sumakant, madam. How are
Yeah,
you?
yeah. Good morning. How are you? Fine. Yeah,
I'm fine.
yeah. So we are very happy to see you.
Thank you. Same here.
Yeah, yeah, yeah. Please carry on. Yeah, please.
Sure.
Thank you.
Thank you. So, Professor Bhaveshri, please let me know when I need to share my screen and when I can start.
Yes, ma'am. A small introduction.
Sure, please.
Then after we'll start, ma'am. Just one more minute. We'll start exactly by 10:30.
Sure.
Working as assistant professor in RTVR Women's College of Pharmacy in the Department of Pharmaceutical Analysis. I welcome you all for the second day of FDP on innovative strategies in drug discovery and therapeutics. I feel immense pleasure to introduce today's speaker, Professor M. Anupama Ma'am, currently working as Associate Professor in the Department of Pharmacy, BITS Pilani. Ma'am has been involved actively in teaching on campus as well as industry professionals for various pharmaceutical industries and independent research since nearly last 10 years. Her research interest and expertise primarily focuses on generating nanotechnology based solutions for diseases like cancer and diabetes. Her lab has been engaged in research activities in the areas of polymers, and ma'am has been published more than 60 research and review articles in peer-reviewed international journals and edited one book and filed 11 Indian patent applications, among which 4 have been successfully granted. Ma'am has been the recipient of prestigious Young Scientist Award in 2015 and Ranbaxy Science Scholar Award in 2011 in Pharmaceutical Sciences. Ma'am also received several awards for the best papers presented at national and international conferences, and her lab has generously funded by several extramural research grants from DST Rajasthan, DST Nano Mission, ICMR, and DBT. Ma'am has guided 4 PhD students and several MPharm students and BPharm students, and she is currently supervising 6 PhD students. And ma'am also serves as one of the founding directors of startup company NanoBrit Innovations Private Limited, aimed towards the translation of nanotechnology based products. With this brief introduction, I would like to uh, welcome ma'am to start today's session.
Thank you so much for that warm introduction. Good morning, everyone. At the outset, I would like to thank Professor Reddy, Professor Sumakant, and Professor Bhavishri for giving me this opportunity to present the research work going on in my lab and to discuss the same with all of you. Let me share my screen. So, in today's talk, I would be mainly talking about two particular active molecules. One of them is a small molecule drug. Its name is lisofilin, and the other one is a peptide. Both of them exhibit anti diabetic action, and how these molecules were difficult to deliver, and hence there was no progress in these molecules. But we started working on them, and the stage that we have reached today has made them deliverable. Before this, I would like to show you our beautiful campus of BITS Pilani from where I come. And this is our classical BITS clock tower, which you might have heard of. Talking about the overall research which is going on in my group, we are mainly working on diseases like diabetes, cancer, AMD, and diabetic wound healing. And we are trying to target treatment of these diseases by using actives like small molecule drugs, microRNAs, peptides, and CRISPR-Cas technologies. Today, as I told you, I'll be discussing the delivery aspect of a small molecule drug and a peptide. So in my lab, we mainly fabricate nano carriers, which are made from polymers, lipids, or a combination of the two, what we call as hybrid particles, to deliver these actives. And we start right from the very beginning, wherein we synthesize polymers to cater to the specific need of these active molecules, which can make these difficult to deliver molecules deliverable, which can protect them from degradation and can prolong their half-life in the blood. So this is one of the structures of the polymer that has been synthesized and characterized in the lab. And then using the polymer and the active molecule, these type of nano carriers are developed, which are further evaluated both in vitro cell culture as well as in animal models. And all the experiments are carried out in the lab itself. We are not outsourcing any part of the experimental work. We truly believe in starting from the very beginning and then translating the innovation in the lab into the benefit for the patients. So today I would be discussing this drug molecule, which was earlier non-deliverable, and we have been able to convert it into an orally deliverable tablet formulation. So, beginning with, we all know that diabetes is a chronic metabolic disease and it's a lifelong disorder wherein there is persistent hyperglycemia in the body. Because of this hyperglycemia, the patients have a very poor quality of life, and over a period of time, diabetes also progresses towards several diabetic complications like neuropathy, nephropathy, and retinopathy. Most of the diabetic therapies are inclined towards use of insulin. However, use of insulin has its own challenges. For example, it alters the gastric emptying time, resulting in postprandial, that is after meals, hike in the glucose levels. It can also result in hypoglycemia. There are problems of weight gain and a high risk of cardiovascular diseases. That is why we are trying to explore certain non-insulin therapies to treat diabetes and its complications. Talking about the first part of my talk, which would focus on making a small molecule drug lisophilin deliverable. This slide shows the background of lisophilin. It is an anti-inflammatory and immunomodulatory drug. We want an anti-inflammatory drug in diabetes because diabetes is considered to be a chronic inflammatory disorder wherein the secretion of several inflammatory cytokines uh, interferes with the normal functioning of the beta cells, and thus the glucose which is produced in the body, it cannot be taken up by the cells and it cannot be assimilated. Lisophilin is also a major metabolite of one of the well-known drugs, pentoxifylline, and currently there have been very few reports which are focusing on developing on this drug for treatment of diabetes. So why we have very few reports? That is because this molecule is very difficult to deliver. Reason being, it undergoes a very fast first pass metabolism, and it has a very short life, half life. It has been reported to be degraded within few minutes in the human body, and therefore it requires a continuous parenteral administration through intravenous or subcutaneous route of administration. Moreover, it has a very high solubility, as high as 60 mg per ml, because of which it is very difficult to encapsulate it in any delivery system. Just to let you know that what is its status in the clinical trials, as I said, because lisophilin, I have abbreviated it as LSF, and we would be using the term LSF throughout the slides to come. So LSF, although it is known to be potent in type 1 diabetes as a standalone molecule, but challenge is with respect to its solubility and high rate of metabolism, because of which, if given by subcutaneous infusion, it is given at a dose of 9 mg per kg, and for IV infusion, its dose is 12 mg per kg, and that too over a 10 hours period. Now, you can understand for a chronic ailment like diabetes, which is not a disorder that requires one time or two-time therapy, rather, therapy is required at least twice in a day. So, going for parenteral phases. Further, we just found one paper on lisofilin wherein some attempts were made to modify the drug itself so as to obtain some desirable properties. So, in this paper, around 32 analogs of the drug have been synthesized, but no in vivo activity of the synthesized analogs have been reported. And since 2006, there is hardly any paper on this molecule. So, lisofilin, if I talk about its mechanism of action as an anti-inflammatory molecule, it acts on a plethora of upstream and downstream targets, which can prevent the incidence of diabetes as well as its progression to diabetic nephropathy. And this slide shows that it acts on so many different molecules, including inflammatory stimuli, inflammatory cytokines. It prevents the release of TGF beta. It prevents lipid peroxide formation and ROS. It prevents the generation of free fatty acids. Accumulatively, all these are factors that contributes towards initiation and progression of diabetes. One of the major mechanisms that have been proposed for the action of lisofilin is by inhibition of this STAT4. By inhibiting STAT4, it prevents T cell activation. T cell activation is important to be prevented because, as we know, type 1 diabetes is an autoimmune disease wherein the immune cells of the body start recognizing our own beta cells as an antigen and start destroying them. So, if we can reduce this T cell activation, it can prevent the destruction of beta cells. In fact, there are several papers which have also reported the activity of LSF in regenerating beta cells. So, with this basic mechanism, the problem with LSF, as I was telling earlier, if we see this is the structure of lisofilin and there is a hydroxy group here. This hydroxy, because of the presence of this hydroxy in this alkyl chain, it undergoes a very quick oxidation and it gets converted into its metabolite pentoxifilin. And this reaction is reversible. Pentoxifilin is also convertible to lisofilin. So this is one of the major problems that we are supposed to overcome if we want to increase the residence time of lisofilin in the body. Our strategy here was if we protect this hydroxy group by conjugating it with a fatty acid, then we would be able to prevent this conversion into the metabolite. And secondly, why we chose a fatty acid was we all know fatty acid is a hydrophobic molecule. One of the major limitations of lisofilin is its high solubility. If we want to increase the residence time of lisofilin in the body, then we would have to conjugate it with a fatty acid. And this fatty acid is lisofilin. Am I audible?
Yes, ma'am.
So I have not been audible right from the beginning.
No, ma'am, just last slide.
Okay, I'll share my screen again. So continuing from where I left earlier, this is my PhD student who mainly carried out the work related to this drug, and presently he is pursuing postdoctoral studies in Canada. Here, our strategy with lisophilin, how to overcome this problem of high metabolism is presence of this OH group is the major reason for its high metabolism and short half-life. And we proposed to conjugate this OH group with a fatty acid because that would protect the OH group and it will no longer be available to undergo metabolism. Why we chose a fatty acid was because it itself is very hydrophobic in nature, and that can overcome the high solubility or high hydrophilicity of this drug. So this was our proposed strategy with which we envisioned that it would reduce the drug metabolism and hence it would increase its half-life. It would also bring down the dose and it has the potential to make the drug orally bioavailable. So, beginning with the research work, as I already mentioned, lisophilin is an anti-inflammatory and immunomodulatory molecule. So, it inhibits the generation of inflammatory cytokines and activation of T cells, and thus it shows its preventive action in type 1 diabetes. We proposed a strategy to formulate a prodrug by conjugating lisophilin with a fatty acid, and the fatty acid was so chosen that it can form micelles. And this is the fatty acid that was chosen, linoleic acid, and the resultant conjugate that was formed was lisophilin linoleic acid conjugate, which was used to prevent diabetes. So this was the synthesis scheme wherein following simple EDC NHS coupling chemistry, we conjugated this fatty acid linoleic to LSF to obtain this lisophilin linoleic acid conjugate, which will be abbreviated as LSF-LA throughout the presentation. Herein, it was purified by flash chromatography, and then by HPLC, we checked the purity, which was found to be more than 98%. This shows the successful synthesis of LSF-LA conjugate. So here in NMR, what was observed, if you see this peak F, this corresponds to an ester peak, which was earlier not present in lisophilin alone. This showed us that successfully we have been able to conjugate the fatty acid, linoleic acid to LSF. We further confirmed this from a C13 NMR. And the expected mass of the conjugate was also verified using HRMS, wherein we got an expected mass of 542 grams per mole. Herein, the molecular weight of lisophilin is 280 grams per mole. And the reason why we chose linoleic acid is because it is a fatty acid with a molecular weight which is similar to lisophilin. It also has a molecular weight of 280 grams per mole. In HPLC, what we observed was because lisophilin is very hydrophilic, it elutes very early in time. Here, if we see, it's earlier than 5 minutes. But when it was conjugated to the fatty acids, the overall conjugate was hydrophobic, and therefore its retention time, it increased to around 18 minutes. Further, we tried to run both the drug as well as its conjugate together, wherein we observed that there was a huge difference in the retention time. This provided an additional evidence that the conjugate has been successfully formed, and as we hypothesized, it is much more hydrophobic than the parent molecule. Next, whenever we are preparing such conjugates, a challenge is whether the conjugate would be able to release the drug in the plasma or not. That is important for the drug to exhibit its action. So for that reason, this conjugate was incubated in rat plasma at 37 degrees Celsius under in vitro conditions. And what we observed was in a period of around 72 hours, the amount of drug that was liberated was around 45%. We could not take this study beyond 72 hours because of the nature of plasma, which itself starts undergoing, which loses its shelf life post 72 hours. Another reason why we did not observe a complete release could have been because we have used plasma which contains metabolizing enzymes. So part of the drug, as it is getting released, it may also have been undergoing metabolism in the presence of plasma. But overall, what we were able to confirm was we have been able to successfully conjugate the fatty acid and the drug. And secondly, the drug is also getting released in free form in the plasma. Next, with this LSF-LA conjugate, as I told you, both had similar molecular weights. So the rationale of selecting similar molecular weight is to form this type of a nanostructure, which we call as micelles. For formation of micelles, it has been well accepted. Then when the hydrophobic and hydrophilic segments are in 1:1 ratio on the basis of molar weight, there is a high probability of forming micelles. If hydrophobicity is more, then it can form nanoparticles. But nanoparticles are not self-assembling in nature, unlike micelles. And we wanted a system which can, by default, by itself undergo self-assembly. So this conjugate, when it was formulated, it was able to undergo self-assembly without using any external surfactant, and we obtained these type of micelles. The size of these micelles was around 156 nanometer with a zeta potential of -17. Because this was a self-assembled system and because micelle formation was observed, in such systems, we also need to determine the critical micelle concentration, which was found to be around 1 microgram per ml. And the N aggregation, which means the number of monomers which are joining together in a single micelle, they were around 54. We also studied the interaction of this LSF-LASM. This means lysophilin linoleic acid self-assembled micelles with a model protein BSA, wherein we found that it did show some interaction with BSA and it had a tendency of binding with BSA. Based on that, we also determined binding constant and the number of binding sites. This LSF-LASM was further evaluated in cell culture studies. The first objective was to see if this is toxic or not. For this purpose, we took insulinoma cells which have a tendency of secreting insulin. And for this purpose, we got MIN6 cells from NCCS Pune. And then this conjugate at different concentrations, 10, 20, and 40, it was incubated with those cells for 48 hours. We observed a near equal viability to 100%, which was seen in case of free drug as well. This ensured that the conjugate was safe to be used with cells. Further, next, for showing any action, the conjugate has to be internalized into the cells. And again, the conjugate was incubated with the cells for 48 hours. We observed that the conjugate showed a very high cell uptake as compared to the free drug. This is attributed to the nano size of the self-assembled micelles. As I showed you, the size is around 156 nanometer. Then what we did was, these MIN6 cells, we induced inflammatory conditions by using a cocktail of cytokines comprising of IL-1 beta, IFN gamma, and TNF alpha. These cytokines are used in a particular concentration and then they can generate inflammatory conditions. That could be seen over here. This is the normal control, normal cells in media, but in the presence of cytokine, CYT is in the presence of cytokine, there is a reduction in the viability of cells because these cause cell death. However, when it was conjugated with LSF-LA conjugate, we see again the cell viability from 40%, it rose to 80%. This showed that the conjugate was able to protect the cell from getting damaged in the presence of inflammation. This was carried out to mimic the inflammatory conditions in the body during diabetes. Further, we also saw if it can increase the production of insulin. So under normal conditions, insulin is at basal level, but the amount of insulin gets stimulated once we take meals, what we see postprandial. So if you see, we checked the effect of the conjugate at both on basal as well as on stimulated levels of insulin. And here we observed that while the normal levels were quite high, it got severely depreciated in the presence of inflammation, which we see in CYT group. But in the presence of LSF-LA conjugate, both at the basal level as well as at the stimulated level, we observed a significant increase in the amount of insulin. And then cytokines, which are inflammatory in nature, these are the ones which are responsible for aggravating diabetes and leading to diabetic complications. If we see here, these cytokines were induced in the presence of a inflammatory modulator PHA. What we observe here is in the presence of PHA, the production of cytokines, both IFN gamma and TNF alpha, is very high. It's around 85 picogram per ml, but it gets very significantly reduced in the presence of conjugate. And this is because the LSF, it shows an anti-inflammatory action wherein it reduces the production of IFN gamma and TNF alpha. Further, we took this conjugate for animal studies. In the animals, we performed pharmacokinetic analysis. We injected the drug at a dose of 25 mg per kg. And we also injected the LSF-LA-SM at the same dose. And what we observed were these two graphs. This was given intravenously, and this black solid marker points, this shows free drug, and the hollow, these diamond-shaped markers, these are showing LSF-LA. If we talk about the changes in the pharmacokinetic parameters that we observed a 5-fold increase in T-half. From merely 0.6 hours, it rose to almost 4 hours after conjugating the drug. And this is the data directly in the animal body. Likewise, there was also an increase in the mean residence time and a reduction in the clearance. Now, if you also focus on this graph, which is in the inset, this shows PTX. PTX is metabolite of lisophilin, which is pentoxifilin. As I mentioned, LSF has a tendency to undergo conversion into pentoxifilin. If you see the solid black marker points, this shows that PTX, which was generated when LSF was administered. And the other graph, which is hollow triangles, this shows that PTX which was generated when LSF-LA-SM were injected. And you can very clearly make out there is a very significant reduction in the amount of metabolite which was obtained from the self-assembled conjugate of the drug. This was further studied in streptozotocin-induced diabetic mice model, diabetic rat model, and herein what we saw was how it impacted the fasting glucose level. Here, another interesting observation was when we started this study, we tried giving both LSF as well as LSF-LA self-assembled micelles at a dose of 25 mg per kg. Intraperitoneally. We got this dose from the literature reference. But what we observed over a period of time was when LSF-LA-SM, self-assembled micelles, were given at 25 mg per kg, the animals started suffering from severe hypoglycemia. Considering that this might be because of overactivity of the drug at this dose, we again repeated the study with a reduced dose of LSF-LA, and we reduced it to 15 mg per kg once daily. LSF-free was recommended at 25 mg per kg twice. And these were the results. If you see the first plot, it is the diabetic control to which no treatment has been given. In between these two plots that you see, hollow circles and the triangles, this is LSF given at two different doses, 25 mg per kg twice daily and 15 mg per kg once daily. Here, one can clearly see that LSF is not able to produce a stable response. The levels of the glucose are fluctuating. But when we talk about the lowermost graph, which corresponds to LSF-LA-SM, given at 15 mg once daily, what we observed was after fourth dose, it was able to stabilize, not only reduce, but also stabilize the glucose levels. We also studied the levels of cytokines in the body and found a significant reduction in the levels of IFN gamma and TNF alpha in the rat blood. This was the histopathology which showed If we compare to the normal control, we see enough of blue colored cells, which are the beta cells which are present in the islets of Langerhans. And if you see here, DC stands for diabetic control, the diseased model without any treatment, wherein there is significant reduction in these cells. Compared to this, if I compared with LSFLA conjugate, the one that was prepared in the lab, you again see a significant rise in the number of beta cells, which is higher than what we observed with the free dose, with the free drug at 25 mg and 15 mg. Again, we observed that even the levels of insulin were way much increased as compared to the diabetic control group or the diseased group. And likewise, we also quantified the insulin staining area. We specifically went for immunohistochemistry of the insulin. And if you see, this brown color denotes or signifies the area which has insulin producing cells, which you see is significantly diminished in diabetic control. However, if you see here, the area has been significantly increased. And when these areas were quantified by ImageJ software, we found the most significant increase in LSF-LA as compared to the free drug. So with this first study, we did conclude that we have been able to prepare a self-assembled conjugate of LSF and a fatty drug. And this has been successful in reducing the metabolism and increasing the half-life by almost 5-folds. Not only this, as we hypothesized earlier, we were able to bring down the dose from 25 mg per kg twice daily to 15 mg per kg once daily. And there was a reduction in the amount of metabolite which was also produced. This work is being protected by an Indian patent. But question was, what next? Since I told you in the beginning, our aim is to translate it to bedside for the benefit of the patients, we wanted to see whether the system is viable orally, because till now, all these studies were being conducted by intraperitoneal route of administration. However, we failed badly when we tried to deliver these micelles orally. It was tried to be delivered orally using an oral gavage, which is a tube that is inserted into the throat of the rat, and then we try to administer the desired amount of the formulation. Here, if you see carefully, this is an oral administration at 10 mg per kg, and the lower two graphs shows pentoxifylline and the upper two graphs shows lisofylline, and one is for free drug, other is for the conjugate, but you see almost a complete overlap. So this conjugate was not able to show any oral viability. The probable reason for this might be this conjugate had an ester bond between the drug and the fatty acid. Orally, in our gastrointestinal tract, there are several esterases which are present, which can easily break or cleave this ester bond. Because of this cleavage, it is possible that within the GIT itself, LSF and LA were cleaved apart, and LSF was again subjected to the same metabolism. This study also shows that how the results which we see under lab conditions vary significantly when we go to in vivo. And we didn't want to take the intraperitoneal route of administration ahead. We wanted to make it orally deliverable. That is why our next step was to formulate a drug delivery system for these LSF-LA self-assembled micelles.